Okay, so after we fixed all technical issues, thank you for that, Zoe. Thank you, Patrick, for the kind introduction, and I'm very happy to be here and present to you one story from my PhD project. And I want to talk about how we developed a novel series of allosteric SHP2 inhibitors, starting out from a scaffold found in orthosteric SHP2 inhibitors. I'd like to start by giving a quick, sorry about this. Okay, so I'd like to start by introducing you to the protagonist of the story, and this is a cytosolic protein tyrosine phosphatase, SHP2. And SHP2 is a key mediator of receptor tyrosine kinase signaling, and its activation can promote oncogenic signaling pathways such as the Ras-Roth-Berg signaling pathway. SHP2 also contributes to the immune evasion of cancer cells through programmed death receptor signaling in T cells. So you can easily see why SHP2 attracts a lot of interest as a potential anti-cancer drug target. Let's move on and take a look at the molecular mechanism at which allosteric and orthosteric SHP2 inhibitors, which are potential anti-cancer drugs, work. So for this, you need to understand that SHP2 cycles between an auto-inhibited closed conformation and an activated open conformation. So in this auto-inhibited conformation, which you'll be hopefully able to see in a second, here it is. And the catalytic site in this catalytic protein tyrosine phosphatase domain is actually occluded by the N-terminal SH2 domain, a regulatory domain that covers the catalytic site. Then upon the binding of the phosphotyrosine peptide to the regulatory SH2 domains, these regulatory domains detach from the catalytic domain, rendering the catalytic site available for substrate binding and catalysis. Our lab at the Leibniz Institute had a long-standing interest in the development of orthosteric active site binding SHP2 inhibitors. But a real breakthrough in the development of SHP2 inhibitors with drug-like properties was only achieved in 2016 when Novartis published this compound, SHP-099, the first allosteric SHP2 inhibitor. And how this compound works is it binds to an allosteric binding pocket and makes contacts to both the regulatory domains and the catalytic domain. And by this, it stabilizes this auto-inhibited closed conformation. The discovery of this allosteric pocket has really reignited interest in this protein target that had previously been deemed undruggable. But now, after the discovery and report on SHP-099, a whole wave of allosteric SHP2 inhibitors binding to that same pocket was disclosed, as you can see from these publications and others. Now, what caught our interest about these allosteric inhibitors is that there is this recurrence of some structural features in almost all published allosteric inhibitors that bind to the site, primarily in this aromatic moiety and in this conformationally constrained amine. So given this recurrence of structural features in allosteric SHP2 inhibitors, we wondered, can we switch the mechanism of inhibition of this N-aryl-1H-azaindazoles core from our orthosteric SHP2 inhibitors to allosteric inhibition by simply exchanging its peripheral substituents with the privileged allosteric site targeting substructures reported in literature? We went to the lab and were delighted that this compound, one of the first that we synthesized, indeed seemed to inhibit SHP2 via an allosteric mechanism of inhibition. And this is evidenced experimentally by the ability of this compound to inhibit the full-length SHP2 protein, but showing no inhibitory activity on a truncated SHP2 construct that only comprises the catalytic domain. That's where the allosteric pocket is not present. This compound, too, was then the starting point for a structure-activity relationship study that we'll focus on during the upcoming minutes. So I first introduced different immune substituents, and this is just a selection of the variants that I tried. But in line with the high conservation of this immune moiety in reported compounds, most of the substituents that I introduced were inactive. Only a few of them showed moderate inhibitory potency, and none was superior compared to the spiroidine that we originally found in the patent barricade therapeutics, and that was then retained for all upcoming designs. Regarding the N-aryl substituent, and here I should point out that the direct N-aryl linkage is really what is novel about our compounds and distinguishes them from the previously published allosteric inhibitors. So regarding the N-aryl substituent, it first seemed that the dichlorophenols motif, which is maybe I can just continue speaking here. So the dichlorophenols motif that is found in most allosteric SHP2 inhibitors published seemed to be most potent among the variants that we tried. But we were surprised to see that also this benzodiazole, which has not been published in the context of allosteric SHP2 inhibitors before, seemed to bring forth good inhibitory potency. But at the heart of my structure-activity relationship work was focusing on the number and position of nitrogen atoms in the N-heterocyclic core. And here we encountered some really surprising activity cliffs and generally a very steep SAR. So just to point out a few examples, if we remove a single nitrogen from this azaindole, leaving us with a mere indole, we dramatically reduce the inhibitory potency of the compounds. Shifting the nitrogen from the 7th position to the 4th position increased the inhibitory potency, and we were also very happy to see an additive effect of the nitrogens in 4 and in 7 position. Most strikingly, when we changed the scaffold from the N-aryl indole to N-aryl-indazole, so just adding another nitrogen in the pyrrole in the right heterocycle, we almost consistently gained about 1 order of magnitude in inhibitory potency. So mind you, this is just changing one single carbon to a nitrogen atom, and it has this dramatic effect on inhibitory potency. Also here we see the same trends. We seem to require a nitrogen in the left ring, and the nitrogen in 4 position is more beneficial than nitrogen in 7 position. And of course, after obtaining this data, we wondered what happens if we combine these two. Well, I'm sorry to say that I have not obtained bioactivity data on these compounds yet, but I'm hopefully soon be able to present these to you. Okay, for the most active compounds, all belonging to the indazole series with 2 nitrogens in this 5-membered ring, we first confirmed their allosteric binding mode by testing them against the SHP2 construct that only comprises the catalytic domain. And as you can see, they were inactive on this truncated version of SHP2, confirming that the inhibition that we see with these compounds is solely due to allosteric binding. The compounds were also tested for their ability to inhibit ERK phosphorylation, a downstream signaling event of SHP2, in an esophageal and a pancreatic cancer cell line where they showed moderate inhibitory potency. They also inhibit with moderate potency the proliferation of an acute myeloid leukemia cell line, but I should point out that the cellular potencies are not at a level where we'd like them to be. Finally, our collaborators at the Max Delbruck Center were able to solve co-crystal structures for some of the most active compounds. And from this representative structure in complex with compound 3, we see that it binds to the same tunnel allosteric pocket as SHP099 and other previously published compounds. It engages residues from the catalytic domain and the C-terminal SH2 domain, as well as the interconnecting loops. The dichlorophenols substituent binds to this rather lipophilic region of the pocket, and here it engages in a cation pi interaction with the guanidine group from the arginine 111 side chain. Potentially, the pyridyl nitrogen here also engages in a hydrogen bonding interaction with this guanidine group, and this could explain why we lose so much potency if we remove the nitrogen from this position in the ring. At the opposite, more solvent-oriented side of the binding tunnel, we find the spironidine-piperidine where the benzylic amine group serves as an affinity anchor by forming 3 hydrogen bonding interactions with surrounding backbone carbonyl groups. Let me summarize what I explained to you. Starting out from allosteric, that is active site targeting SHP2 inhibitor, we discovered a novel series of N-aryl-1H-azaindazoles and azaindazole-based allosteric SHP2 inhibitors. The most potent derivatives from this novel series of allosteric SHP2 inhibitors have nanomolar inhibitory potency on the isolated enzyme and inhibit downstream phosphorylation of the ERK protein in cancer cell lines. Currently, the evaluation of some of the most active compounds is ongoing in vivo, and in the future, we are very interested to explore if a synergism can be observed on a combination treatment with orthosteric and allosteric SHP2 inhibitors, because we believe that this could be a strategy to tackle activated SHP2 mutants, which are generally less susceptible to allosteric SHP2 inhibition. Now, before ending my presentation, I'd just like to take 1 minute to introduce you to another EU project, an open science initiative, and that is EU OpenScreen, who our group at the Leibniz Institute in Berlin collaborates with closely. So if you're a synthetic chemist or for any other reason have some old compounds lying around in your freezers, you might consider donating them to EU OpenScreen's screening library. This library is regularly screened against a broad range of therapeutic target and different assay systems. And by including your compounds in the screening library, you could explore novel bioactivities of your compounds and contribute to the scientific community. If your compounds would pop up as hits in these screening initiatives done by EU OpenScreen, they would of course inform you on the bioactivities of your compounds. They could also connect you with potential collaborators who are interested in your molecules. So yes, if this sounds exciting to you, you could either approach me with any questions or contact directly Victoria Mora and consider donating your compounds to explore novel applications of those. Okay, and with this, I would like to thank first of all my PI, Mark Nazari, who's also here today at our lab in Berlin for providing a really great working atmosphere. I'd like to extend thanks to all collaborators who contributed to this project, and of course the organizers of the conference and the ALLODD consortium for supporting my PhD project over the past years with really nice meetings, insights, tips, discussions, and of course, for organizing this event. I'm very happy to be here and happy to discuss with you during the break or now if you have any questions. Thank you.
Yes. Question. Yeah. My question is because the standardized approach of will be controlled or
Yes, so it's exactly as you proposed. These molecules bind to a site that lies at the interface of the regulatory domains and the catalytic domain. And by engaging residues from all 3 domains, they stabilize this auto-inhibited conformation. So you could think of them as like intramolecular glues, so to say. They tether the regulatory domains to cover the catalytic site.
Other questions? Yes.
Maybe I missed it, but even that your crystal structure shows that in H you engage in so many interactions. Did you try the and see that we keep that?
Thank you, Jordi, for this question. It's something that has been suggested by me quite often already. So yes, I have synthesized the enantiomer. Indeed, it shows lower inhibitory potency. Not as stark as during the previous talk, but there definitely is an effect, yes.
also a question. We can go one slide back. This one, yes. Maybe is there also a possibility, because you crystallize it now without the CNN SH2? Is there also the possibility to block the mechanism directly at the interaction interface of these two LNC terminal parts? Because there's no crystal structure of an active form, what I saw, but maybe you can block this also directly at the interaction site. Is it also possible?
You mean in the interface between the N
Yes.
SH2 domain?
For example.
Maybe Vincenzo will allude to this during his talk. I don't know if your compounds make contacts with the NSH2 domain. As far as I know, there is this allosteric site and then another one that is located around here, but I'm not sure if the inhibitors really form interactions with residues of the NSH2 domain. And yes, to my knowledge, indeed, there is no co-crystal structure of the open state comprising these regulatory domains. Yes.
Other questions? If it's not the case, then thank you very much for the very nice presentation.
Thank you.